So guys, today in this video we'll be talking about the body's neural system, one of the most complex systems of our body. Here I'm drawing brain together with the spinal cord, and these two makes the central nervous system of our body. As these structures are very important, these are enclosed within the bony cavities. The brain is enclosed within the cranium and the vertebral column encloses the spinal cord. Here I am drawing the limbs of our body and the trunk. The limbs of our body have the bone and the muscle which facilitates the movement of our body, and the trunk contains the visceral organs, the heart and the lungs. So this is the brain and this is the spinal cord. The brain and the spinal cord, they receive sensory impulses from the periphery with the help of nerve, and they send motor commands towards the periphery also with the help of nerves. So these nerves together make the peripheral nervous system. Now the peripheral nervous system consists of nerves, and here I'll be drawing the nerves in blue. So these nerve fibers are basically of two types: the afferent sensory fibers and the efferent motor fibers. The sensory fibers bring the impulses towards the central nervous system for processing, and the central nervous system sends information or the motor command to the skeletal muscle and the visceral muscles with the help of the efferent fibers. Whole of the neural system, that is the brain and the spinal cord and the peripheral nervous system, they consist of structural and functional units which are called the nerve cell or the neuron. Billion of these neurons are present inside our body. The brain alone contains 100 billion of these neurons. So if we were to zoom in and try and look into one of these neuron, it would look something like this. So here I would be drawing the structure of a neuron. Now the neuron consists of the cell body or the cell soma. From this, a number of these projections are arising which are called the dendrites. So these projections arising from the cell soma are called the dendrites. And finally the cell soma lead into a long fiber which is called the axon, and here I am drawing the myelin sheath. This myelin sheath may or may not be present around the axon. This axon then ends into what are called the axon terminals, which end into synaptic nerves, which are so called because they participate in the formation of synapses. So this is the whole structure of a neuron. And here I'll be drawing the cell nucleus present in the cell soma. So this is the structure of a neuron. These are the dendrites which carry impulses towards the cell soma, and the axon carry impulses away from the cell soma, and they end into synaptic knobs. So this is the cell nucleus. Here is the cell nucleus, and in the cell soma are present a number of these granules also in the dendrites, and these are called the Niesel's granules. These are basically the remnants of rough endoplasmic reticulum, and they take part in the formation of proteins, and these projections are called dendrites, which bring sensory or which bring information towards the cell soma. So this is the cell soma, which contains the nucleus and the Niesel's granules and a lot of other organelles. This is the long fiber axon, and this is the axon terminal, and this is the synaptic nerve. And this what I have drawn is a example of myelinated nerve fiber. So sensory, the information is brought to the cell soma by dendrites, and this information is carried away by axon and passed on to the next neuron through the formation of synapse. Now if we were to take a cross section across this nerve fiber and look at it, it would look something like this. Now I'll be drawing the axon fiber in black. So this in black is the axon fiber, and around it, it is a myelin sheath which consists of concentric layer of this myelin deposited around the axon. So these are concentric layer of myelin deposited around the axon. This myelin sheath is basically formed by the Schwann cell, and we will look at how it is formed. This outer covering of axon is called the axolemma, and the outer covering of the whole neuron is called the neurilemma. So how this myelin sheath is formed? So this is formed by the Schwann cell. Now here again in black I'll be drawing the axon, and around it is present a Schwann cell. So this is one of the Schwann cell, and the Schwann cell has this nucleus which I have drawn now. So this Schwann cell tends to grow and wrap around the axon. So in the next step, one of the process of this Schwann cell will grow, and spirally it will grow around the axon and it will cover it like this. A number of when a number of these concentric turns are made, concentric myelin sheath is formed around the axon. Now when we talk about the nerve fiber, we say that they carry sensory information from the periphery towards the central nervous system, and this carry the motor commands from the central nervous system towards the periphery, that is the muscles. So how this sensory information and the motor commands are carried by the nerve fibers? They are basically carried in the form of nerve impulse. Now to understand that, let's take a part of this axon fiber and zoom into it and look at it. So here I'll be drawing a part of the axonal nerve fiber. So this is a part of the axonal nerve fiber which I am drawing now. Now axonal nerve fiber contains a number of ion channels. These may be leaky channels. By the word leaky channels, we mean that they are always opened at resting condition, and therefore they are called the leaky channels. And they also contain the gated channels, which only open in the presence of a stimulus. These gated channels may be voltage gated channel, ligand gated channels, mechanical gated channels. So basically, these are the gated channels which open in presence of stimulus, and there are leaky channels which are always open. So here I'll be drawing one of the leaky channel. This is basically the potassium leaky channel, and this is the sodium gated channel, which is normally closed in absence of a stimuli. So potassium ions can go outside from the leaky channels, and the sodium ions at resting condition cannot come in with the help of these gated channels because they are closed now, and they contain this ATPase pump which pumps out 3Na plus outside and 2K plus inside. And inside of the nerve axonal membrane are present these negatively charged proteins. So inside are present negatively charged proteins. 3Na plus is going outside while only 2K plus are coming in. That is, more positive ions are going outside and less positive ions are coming in, and a lot of negative ions are already present. So all of this makes the inside of the axonal membrane relatively negative in comparison to outside, whereas the outside becomes positive. This is the formation of two poles. Therefore, the axonal nerve membrane at resting condition is said to be polarized. The formation of two poles, positive pole outside and the negative pole inside. So the nerve fiber at resting condition is in the state of polarization. So this is the resting condition of the axonal nerve fiber. Now when a stimulus comes, now here I'll be drawing again axonal nerve fiber. Now in the presence of stimulus, these gated ion channels, the sodium gated ion channels which were closed, they will open up. So these gated channels will open up. Now when these gated channels open up, a lot of these sodium ions which were present in excess outside, they start coming in. So a lot of these sodium ions are coming in. As a lot of the sodium ions are coming in, the inside of the membrane which was present, which was previously in the negative state, the negative charge will be negated by the incoming positively charged sodium ions. So this is again this is becoming depolarized. The poles which were present are becoming, are going away. So this is was present in the state of polarization, and now this has become depolarized. Now if we were to show this in the formation of, in the form of a graph, so here I'll be drawing a graph. In the y-axis we will be representing the voltage. So this is voltage on the y-axis, and on the x-axis we will be representing time. So the nerve fiber at resting condition is present in the state of polarization, and the inside is negatively charged. So inside maybe let's say minus 70 millivolt. Now when the stimulus comes, this voltage gated channels open up, and a lot of the sodium ions start coming in. So inside becomes positive and it goes towards the positive side. So this is called, what is called depolarization. And this depolarization is because of the influx of the sodium ions which came because the voltage gated channels opened up. Now this disturbance which was produced, the depolarization, the nerve fiber will again become polarized, and this, that is called repolarization. That is when the voltage of the, when the side becomes positive, this voltage gated sodium channels close up. So this will be closing up, and a lot of this potassium gated channels will open up, and a lot of these potassium ions will be going from inside towards the outside of the axonal membrane. And as the inside loses a lot of these positively charged potassium ions, the inside will again become negative because it has lost a lot of positive ions, and the outside will become positive. So this is what is called repolarization. Again the two poles are formed, positive and negative poles. So this is repolarization. This is because of the efflux of the potassium ions, and this is called repolarization. And then again the resting condition. So this disturbance due to stimuli, which is this disturbance when a stimulus is applied, this is what is called the nerve impulse, and it is shown in the form of this graph which is called the action potential. And the this disturbance is then again propagated towards the whole of the neuronal membrane. How that is propagated, this action potential of the nerve impulse which was produced at one side of the axonal membrane when a stimulus was applied, how this nerve impulse which was produced, or the disturbance which was produced in the axonal membrane, how this disturbance of the action potential or the nerve impulse is propagated throughout the whole nerve, whole of neuron. We will be studying that in the next video, and we will also be studying how this uh, nerve impulse is then carried to the next nerve cell through the synapse. So we'll be talking about that in the next video. So that was all about today.